Welcome back. Welcome to part two. We continue our discussion on development and especially the Hox genes. For instance, the Hox genes that are in the regions that make the head are going to direct things like the mouth parts and antenna to grow, and different parts of a fruit fly like a thorax are going to direct things like the legs or the wings. Um, the fruit flies have 8 Hox genes, and they're all on the same chromosome, located on chromosome 3. And what's interesting about Hox genes is they typically are found in the same order on the chromosome as they are in the body plan. As here, Hox gene 1 develops these parts, and then as it goes back, all the Hox gene 9, and that's the general body plan of the fruit fly, which is interesting. This also just shows that how the thorax segments are here, the head segments are here, and this is a normal one. A lot of what these scientists did was they studied this, but with mutations. Here's an interesting one. Where antennae are supposed to be growing, you have legs. That's because scientists can take certain Hox genes and move them around, and get different expressions of body plans, which is really kind of crazy, and it helps show them, what Hox genes did and how they control certain things. Got to go through this here kind of quick. Nearly every animal that's studied has shown to have the homeobox sequences or the Hox genes in its DNA, suggesting that Hox genes are very ancient in the early animal family. And certain Hox genes have been duplicated, and they seem to be very similar. And you can see that here. You got, one Hox gene's been duplicated, it's been duplicated, and after it's been duplicated, those Hox genes can then mutate and control different things. It's kind of like this idea of you have a gene that works, one Hox gene, then you duplicate it, and you give that second one, you modify it a little bit to do a little bit different thing, which would maybe enable the animal to grow a different structure to help it survive. And here's another example of different structures that have morphed their Hox genes to do different things, make a stinging tail, make wings, make many legs, things like that. The more complicated you get, the typically you have more Hox genes. Lots of times the whole Hox, like all of this will be duplicated, and you'll get 2 of them. So instead of having 9 Hox genes, you'll have 18. And now you have all these more Hox genes that can do specialized things. That's kind of what's going on there. And so pattern formation, it deals a lot with Hox genes. And then that leads into the morphogenesis, which is the specialization. It might not seem like humans or mice are segmented, but they are. The vertebrae especially are segmented, but even things like your muscles all derive from a segment, or your fingers are kind of segmented, and things like that. So there's lots of segmentation, lots of complexity that comes with this. This slide talks about how neighboring cells often control morphogenesis or cell specialization. Neighboring cells can kick off what's called the MyoD gene, which will eventually lead towards a cascading effect, turning a cell into a muscle gene. And that's kicked off by neighboring cells. Neighboring cells put off factors. Morphogenesis can involve cell death or apoptosis. And you can see how that can create some digits here. This is an example of a normal white blood cell and one going through apoptosis. That can happen because of all kinds of things. This is an example how programmed cell death can lead towards webbed feet or non-webbed feet in a bird or in a duck, because essentially those programs are turned off here. The genes turn off those apoptosis programs to keep webbed feet. That's called a heterotopy. Heterotopy is kind of like a turning off of that. Heterochrony is timing. So in a long a giraffe, you have different, these cells grow, or these bones grow, and they're not turned off. So they get really long versus like a human. We'll